structures. So is there any doubt in the oviduct part and in the uterus part? Everything is clear to you? So is the screen visible to you? So we had started from here. It's visible to everyone. So this is the female reproductive system. Once again, we will just brush up. So this is a closer view of the female reproductive system showing the ovaries as well as all the parts. This was the primary sex organ, a section through the ovary. We have already done the process of folliculogenesis is being shown over here. Again, the various stages of development of the ovarian follicles from the primordial to the Graafian follicle and how they rupture and what they form. Again, this is folliculogenesis, another slide. This is anatomy of the Graafian follicle, the different layers of the Graafian follicle we had already discussed. Ovary we had discussed. Oviduct, I had told you the different parts of the oviduct. Uterus and vagina, I had told you about the different parts of the uterus. Okay, and coming to the vagina. Now, vagina is also known as the birth canal. Vagina is also known as the birth canal that I told you yesterday. And it is lined by the stratified squamous epithelium. Stratified means that it is multiple layered, and squamous epithelium means the cells are flattened. So the vagina is lined by the stratified squamous epithelium. And I told you the vagina contains Lactobacillus acidophilus. This is the bacteria. So this bacteria converts glycogen to glucose. Sorry, it converts glycogen to lactic acid. So it converts glycogen to lactic acid and thereby lowers the pH. So look at the pH, it becomes low, and because of this, it acts like it protects the vagina from the attack of pathogens. And then we had studied the uterine structures. There was this blood supply to the uterus. You can see the aorta that branches. There's this common iliac artery that branches into external and internal artery. The internal artery is the artery that supplies the female reproductive system. It branches the uterine artery goes into supplies the wall of the uterus. So it further branches to form the arcuate artery. Arcuate artery, all this is taking place in the myometrium. So they are supplying to the myometrium, and arcuate artery branches to form the radial arterioles. And just before entering the endometrium, they become spirally coiled. So they are called spiral artery. And because of this, there is a limit of the days in which the blood flow takes place. Because as the arteries are broken during the menstrual flow, they again get coiled and mend themselves. So this is how the bleeding is not continuous. Understanding? So this is a closer view of the various arteries in the wall of the uterus. Yes, and coming to the female external genitalia. So I think we had stopped over here, the female external genitalia, and I told you the female external genitalia consists of the mons pubis. So this is an elevated area made up of adipose tissue. So mons pubis is an elevation of adipose tissue. I told you it's an elevation of adipose tissue, and it is covered by skin and pubic hairs. It is covered by skin and pubic hair. So all over, this is the mons pubis. Then you have two important structures. You have two important structures. The larger fold called the labia majora. The larger fold called the labia majora that is present externally, and the inner fold labia minora. So you have this bigger fold, external fold, and then you have this smaller internal fold, which is labia minora. So mons pubis elevated area, larger external fold, labia majora, and the inner fold, which is labia minora. And the space enclosed by the labia minora is the vestibule. You can see the vestibule is labeled here. This is also an external, female external genitalia. And vestibule has a urethral opening and the vaginal entrance or the vaginal orifice. So this is only covered with hymen. And hymen is a porous structure. Blood can flow through the hymen. All right. So these are the important structures. Now, you also have two important glands. You have two important glands on the sides of the this is the on the vaginal near the, you can see on the margin near the labia minora. This is the greater vestibular gland. This is the greater vestibular gland. And what is the other name of the greater vestibular gland? Can you tell me? I told you yesterday what is the other name of the greater vestibular gland? Anybody knows the other name of the greater vestibular gland? It is the Bartholin's gland. It is the Bartholin's gland. So where is the Bartholin's gland? It is the greater vestibular gland. All right. The greater vestibular gland is called the Bartholin's gland. And what is the function of the Bartholin's gland? What is the function of the Bartholin gland? The Bartholin gland secretes a mucus-like substance. So this is the greater vestibular gland, or called the Bartholin gland. There is another gland which is called the, and this Bartholin gland is, like or homologous to the bulbourethral gland in male. You also have a lesser vestibular gland, which is not shown over here. There's another gland which is called the lesser vestibular gland, which is also called the paraurethral gland. So this is near the urethral opening, and while the greater vestibular gland is near the, it is near the labia minora. All right. So near the vaginal orifice, you can say these glands are there, and they produce mucus. Now, you can also see a structure over here that is called glands of clitoris. This is homologous to the glans penis. This is homologous to the glans penis present in male. Homologous means the same tissues form both of them. So all the time you have to know which is homologous to what. So like the Bartholin's gland is homologous to the bulbourethral gland, whereas the lesser vestibular gland is homologous to the prostrate gland. The clitoris, which is homologous to the clitoris, which is homologous to the glans penis in male. Again, I'm going to tell you about all of this. All right. So these are the important structures. These are the important structures that form the external genitalia of a female. External genitalia of a female. Now, you also must have heard about the cervical mucus. Do you have any idea about the cervical mucus? What the cervical mucus means? Cervical mucus, where is cervix? Cervix is the narrowest portion of the uterus. It is present since birth. Okay, so let me just complete this. We were speaking about clitoris first. So all these structures are clear to you. So once again, I'm telling you that the female external genitalia consists of the mons pubis, the labia majora, the labia minora, vestibule, openings of the vestibule, glands, all right, and the clitoris. So clitoris is a small cylindrical mass, you can see. So it is a small cylindrical mass, and it consists of two erectile bodies. It's very small over here, but it consists of two erectile bodies, and they are like homologous to the glans penis, and like the penis has the layer of skin that is also present in the glans of clitoris. So that's why it's homologous to the glans penis, and its main it also helps in its role is in sexual arousal. So the same functions as the glans penis, the clitoris also has the same functions. All right. So this is also glans of clitoris, or clitoris is also an important structure of the external genitalia of a female. External genitalia of a female. So these are the important structures. Now coming to the next structure, which is the mammary gland. Mammary gland is also an important structure in the female that also forms a secondary sexual characteristic in a female. The presence of breast, the presence of mammary glands is very important for a female. Mammary glands are two in number. You can see mammary glands are two in number. And where are they located? Where are the mammary glands located? A functional mammary gland is a characteristic of all the females, of all the female mammals, especially. And they are paired structures. You know that they are paired structures, the mammary glands. And what do they contain? What do they contain? They contain glandular tissue. They contain glandular tissue and variable amount of fat. What do you mean by glandular? Glandular means they have glands that secrete milk. They have glands that secrete milk. And variable amount of fat means the fat content in the mammary gland is not same in every female. It varies. Okay. So the glandular tissue of each breast, mammalian breast, The glandular tissue, now this is one portion of the mammary gland magnified. So the glandular tissue of each breast is divided into 15 to 20 mammary lobes. So there are 15 to 20 mammary lobes in each breast. Okay, and where are those lobes? You can see over here. Now see, I told you they are paired structures and they contain glandular tissue. Glandular tissue, that means they are made up of fat. They are made up of fat. And where is the fat over here? These yellowish structures are the fat lobules. Everywhere you can see. So the fat content varies. The fat content varies. Yes, it is a modification of the sweat gland. Sweat glands are mammary glands are modifications of sweat glands, and you can see you can see variable amounts of fat. Now each mammary gland is made up of 15 to 20 mammary lobes. 15 to 20 mammary lobes. So where are the lobes over here? It is divided into 15 to 20 mammary lobes. So these are the mammary lobes. These, the bluish ones, you can see. So these are the lobules or lobes. And what is there inside the mammary lobules or lobes? Inside the mammary lobules, you have clusters of cells called the alveoli. So if these are the mammary lobules, inside what you have is clusters of cells called the alveoli. So you can see in this diagram that these are the mammary glands, and inside the mammary glands, you have these alveolus or alveoli. Alveolus is singular. So alveoli word, where have you heard? You've heard while we were doing the lungs, right? And what is the function of the alveolus? The cells of the alveolus secrete milk. And this milk is stored in the cavities of the alveolus. For example, if this is one alveolus magnified, so you will have milk which is being produced inside the alveolus and stored in the alveolus. Okay, and the alveolus opens into mammary tubules. So if there are many alveolus, and all of them have a duct, and these ducts open into mammary tubules. You can see this way. So how is the organization? You have the fat layer all around, then you have the mammary lobes, alveoli inside or alveolus inside the lobes. Then you have ducts coming out from the alveoli that merge and form the mammary tubules. Mammary tubules, each mammary tubule joins to form a mammary duct. So you have the alveolus, you have the mammary tubule, and then you have the mammary duct. So where is the mammary duct? Have they labeled mammary duct? No, they haven't shown. So you have the mammary tubule, then you have the mammary duct. So what emerges from each lobe is the mammary duct. All right. And several mammary ducts, they join to form a structure called the ampulla. Okay. And the ampulla is connected to the lactiferous ducts. So milk is formed in the alveolus travels to the mammary tubules, then to the mammary ducts, travels to the mammary tubule. So milk is formed in the alveolus, travels to the mammary tubules, then to the mammary duct. From the mammary duct, it travels to the ampulla, from the ampulla into the lactiferous ducts. And via the lactiferous ducts unite like this and it opens into the nipple. And nipple is covered with this brownish area called areola. So areola is by the help of this dark structure that the newborn can distinguish this, because the newborn is not able to see anything properly. All right. So this is the organization of structure of a mammary gland. Now, what do you have this side? Coming to this side, look at the location of the mammary gland. Now, mammary gland is present in the pectoral region. So you have ribs over here. So these are the ribs, and you have a muscle called the pectoralis major. And on the pectoralis major rests the breasts or the mammary glands. All right. So this is the location. So if we just want to properly understand, as in what is the association of the, or what is the order of the various structures. So let us just write it once. Mammary glands. There is this alveoli, then there will be alveoli lumen, where milk is produced. From there, it travels to the mammary tubules. That means the various ducts of the alveoli, they form the mammary tubules. Mammary tubules unite and form the mammary ducts. The mammary ducts form the mammary ampulla, and the various mammary ampullas, they join the lactiferous ducts. So this is the organization that takes place. So milk production starts in the alveolus, comes to the lumen, then to the mammary tubules, then to the mammary ducts, to the mammary ampulla, and from here it comes to the nipple area. So did you follow this? Cells inside the alveoli. There are various the cells, secretory cells. They help in the formation of milk in the alveoli. And you know, after they are formed, after the milk is formed, then what happens? There are glands which burst and release their secretions. No, the same thing happens over here. So have you followed this? The order should be clear to you, because in the exam they ask you the order. In the NEET exam they ask you the order. They will just write any order and they will ask you whether the order is correct or not, and you have to identify which is the correct sequence, how the milk is formed and how it travels via the ducts. So that is very, very important, and you should be properly knowing as to what is the path. So that's why this order is very, very important. Please note down this order, and you will require this to do the MCQs. So functional mammary glands are found in all the females. They are paired structures. They are glandular tissues. And there are 15 to 20. Now this number is asked. There are how many lobes? There are how many lobes? So 15 to 20 mammary lobes are found in each breast. This is asked as a question. And each mammary lobe is divided into numerous alveoli, and the alveoli has lumen. So inside the alveoli, you will find milk is being produced. Inside the alveoli, you will find milk is being produced. All right. And then the alveoli open into the mammary tubules that join to form the mammary ducts. Mammary ducts join and open into a dilated area called the ampulla. Mammary ampulla further narrows and continues as the lactiferous ducts that opens at the surface of the nipple. So this information you should be able to understand. All right. So is it clear to you? Yes. What are your doubts? So we are done with this. Now we will start the male external genitalia. That means the male secondary sexual organs. So while studying the male reproductive system, coming back to the male reproductive system, I told you it consists of a pair of testes. This is the primary sex organ. We had done all about the testis and the scrotum. Accessory glands was left, duct system was left, and the copulatory organ was left. So under the duct system, you have epididymis, which is not, you have the epididymis, you have the vas deferens, and glands like prostrate. Then you have the seminal vesicle. Then you have the Cowper's glands also. So there are three glands, three accessory glands, and epididymis, vas deferens, all of these form the duct system. So today's class, we are going to do the duct system and the glands. Okay. Now look at this. This is the structure of epididymis. This is the structure of epididymis. Epididymis, as in you can see, this is the testis. And from the testis, now see, inside the testis, as I told you, the tunica albuginea divides the testis into compartments, and each testis has testicular lobules. They has lobules and seminiferous tubules. All right, and seminiferous tubules then form these straight tubules, which merge and form this. What is this structure? This netted kind of structure. This is the rete testis. This is the rete testis, and here is again a structure which is the vasa efferentia, and from here this green structure that is shown over here is the epididymis. So this epididymis, you have the head of the epididymis, which is called the caput. You have the body of the epididymis, and you have the tail of the epididymis, that is cauda. So what does the epididymis do? What role does the epididymis have? That is very important. You do get questions based on epididymis. So first of all, testis plus epididymis is known as testicles. In the common language, we often refer to as testicles. So testis plus epididymis is called testicle. If you get an MCQs, you should be able to do. Each epididymis is around, it's a long tube, 7 to 12 meters, highly coiled. That's why it fits into this area. It's highly coiled. And what does it do? Why do we require, why the male reproductive system? Why does the male reproductive system, why the male reproductive system require the epididymis? So this is 7 to 12 meters coil, and it is this site for temporary storage of sperms. Sperms are temporarily, so it is the temporary storage of sperms. It is also the site for maturity. Sperms, see, sperms which are released, or spermatozoa which are released from the testis, no, they are not active. They are not active. They are not active. All right. So what happens to them? They are not active. So as these sperms travel from the seminiferous tubules to the rete testis via the vasa efferentia to the epididymis, they are activated by the fluids present inside the epididymis and the vas deferens. So basically they are inactive at this point of time. If you just take out sperms and in vitro and try to fuse it with ovum, they will not fuse because they have not attained that activeness. So this epididymis is a place where the sperms are temporarily stored and they attain maturity and motility, also the ability to swim. They are inactive, not so active. They're just moved on. Okay, and there, while they are inside the epididymis, their nature changes. They become more and more motile, more active. So the pattern of motility and the percentage of motility, that means the ability to swim, the ability to move, changes as the sperms travel from the head of the epididymis to the head to the tail of the epididymis. Now, for what amount of time do they remain inside the epididymis? That is a question. So they remain inside the epididymis up to around, a month or so. And once they are ejaculated, that means once they are out of but the sperms are not active. So in order for the sperms to be active, they travel in the epididymis, which is the temporary storage. Then they travel to the vas deferens, which is the secondary storage. Then they meet the secretions of the various glands. All those secretions get added, and when they are ejaculated out in the female reproductive tract, they developed the ability to swim. So all the time, how do they move? Now you should ask me that, ma'am, how all the time, how are they moving if they are not active? So basically, all the time inside the testis and in the epididymis and everywhere, they move, they are propelled by the fluid movements, the fluid secreted by the ejaculatory duct and the vas deferens, and the peristaltic movement in the vas deferens basically helps in the movement of the sperms. You're understanding? But totally they become active when they reach the female reproductive tract. And over there's a process called, where they are activated and they receive secretions, and they travel to the oviduct. Over here, you will see that the various glands like the seminal vesicle, it adds up various nutritive substances into the semen like fructose. So we will discuss about all that. So what we have done for epididymis is around 7 to 12 meters long. It's coiled, and testis plus epididymis is called testicle. It is the site for temporary storage, maturity, and motility of sperms. Sperms remain in they are stored in the epididymis. But see, what happens is sperm production is a continuous process. All the time ejaculation is not taking place. So what is happening to the sperms? So this epididymis is also a place, is also acts like a recycling unit. The phagocytic cells present in the epididymis, they digest the sperms. Otherwise, always there will be an overflow. Understanding? So this is also a function of the epididymis, is to it acts like a recycling unit. It acts like a recycling unit. So it digests the sperms. It digests the sperms all the time. The sperms are being produced. So the recycling center of the sperms is the epididymis. So all the time phagocytosis takes place inside the epididymis. So understood. So what is the function of the epididymis? The function of the epididymis is to temporarily store the sperms, provide maturity, the ability to move slowly the sperms become active. Also, it helps in phagocytosing the sperms. Okay, so this is the epididymis, the first duct. They can remain in the epididymis for around a month. So they can stay in the epididymis for around a month. And something you should know, that once ejaculated, once the sperm is out of the male's body and is into the female reproductive tract, The total time at till which it can survive is approximately 3 days, 72 hours. But you know, the fertilization should take place within 48 hours. Otherwise, the sperms will die. So it is active once being ejaculated into the female reproductive tract. It is active for around 3 days, around 72 hours. But yes, 48 hours, like 2 days. I mean, after 2 days, they mostly die. And during those days, when they have reached this the female reproductive tract after such a long journey, um, less amount of sperm survive, and because there is difference in the pH of the you know, male and female reproductive tract. So when we do the process of fertilization, I'm going to tell you what are the hindrances and how much, you know, how much effort the sperm makes to travel such a long distance through the female reproductive tract, and what are the barriers it has to overcome. Understanding. So this is about the epididymis. So if they ask you a question, so questions asked over here is which is the temporary storage, which is the recycling unit for sperms. So your answer will be epididymis. Your answer will be epididymis. Now the second accessory structure or the duct is your vas deferens. Vas deferens. Vas deferens, you can see the vas deferens is over here, and vas deferens is around 45 centimeters. So it's like small, not so long like the epididymis. So it is around 45 centimeters in length, and it ascends along the posterior body of the epididymis. So see where the location is. Look at this. So this is, look at this diagram. You can see this is the head of the epididymis. This is the body, and this is the tail, the chorda part. So from here, from the chorda part arises this structure is the vas deferens, okay, or the sperm duct, which you call. So the sperm duct arises from the posterior body of the epididymis. So this is the posterior part. So this is arising, this vas deferens is arising from the posterior most part. And where does it go? It goes into the abdomen, right? So look at this diagram, it will be clear to you. So these are the testes, and this is the epididymis. You can see the posterior border. So this is covering the vas deferens's location is posterior border of the, touches the posterior border of the epididymis. And then I told you about spermatic cord, right? This is the spermatic cord. Spermatic cord is the cord through which the testis remains suspended. The scrotal sacs are suspended through the spermatic cord, and this is the inguinal canal, the passage through which it comes out. The scrotum comes out, right? So it takes the same route. The vas deferens take the same route. So they ascend along. They ascend. That means going up. Descend means coming down. Ascend means going up. So the vas deferens ascends along the posterior body of the epididymis through the spermatic cord, and then enters the pelvic cavity. So it is entering the pelvic cavity. Now inside the pelvic cavity, it makes a loop over the ureter. It makes a loop over the ureter. You have the ureter, all right? So it makes a loop over the ureter and then passes over side and down the posterior surface of the urinary bladder. Just a second. Let's look at this diagram. Where is the, this is the vas deferens. Can you see? So it is making a loop over the ureter. It is making a loop over the ureter and passes side and down through the posterior surface. And then, you know, over here, it broadens to form a region called ampulla. So this is also a place where sperms are stored. So this is the site for secondary storage. And then as it moves down, it meets the duct of the seminal vesicle. And these two together, where the duct of the seminal vesicle meets the sperm duct or the vas deferens, it is called the ejaculatory duct. Understood? So what all we have mentioned about, or which is significant about the vas deferens. The vas deferens, (45 centimeters in length, loops over the ureter and passes to the posterior surface of the urinary bladder. Okay, and the dilated portion of the vas deferens or the ductus deferens is ampulla. So they ask you about this ampulla. Ampulla is also present here. This is the secondary storehouse of the sperms. So which is the primary storehouse and which is the secondary storehouse is important. All right. So this is the secondary storehouse of the sperm. Look at this diagram carefully. What are they showing? They're showing testes, then they're showing epididymis, then see how the vas deferens is arising and moving into the pelvic cavity, and then it will loop around the urinary bladder. That I had shown in the next diagram. Can you see this portion? From here, a small portion of the vas deferens has been cut and these ends are ligated. So this is the surgical method of birth control, which is called vasectomy. So once the vasectomy is done, so the sperms are produced, but they do not, the semen is produced, but it does not contain sperms. But this technique is not active, like, just you've done and, it's sperms are not going to be there. It takes some amount of time. So that, because the sperms are present here also. So all these sperms should get exhausted. So after being made, they are stored. I told you this is the secondary storehouse. So sperms are being stored. So till all the sperms over here are exhausted, it cannot be a birth control measure. So you have to wait after a vasectomy done. If a couple does not want children, then they have to take care of this, that all the sperms in the upper portion of the vas deferens should be exhausted. All right. So understood epididymis and vas deferens. If they are not ejaculated to the female, so they are being they die, and there's this process of phagocytosis which is taking place in the epididymis also, and also in the some amount is taking place in the vas deferens, but primarily in the epididymis. I told you, the epididymis is the primary recycling unit. I mean, where due to phagocytosis, the sperms are digested also. So all the time, no, they are digested. That means the phagocytic cells will come and engulf them. No pain. All right. Now look at this diagram. This diagram shows accessory struct glands in the male, and there are 3 important glands. You can see a pair of seminal vesicles, and then you can see ejaculatory duct where the sperm duct meets the seminal vesicle. This is a gland called the prostrate gland. And then 2 other glands which are in a pair. This is the bulbourethral, one on each side, or the corpus gland. So you can see that this is the urinary bladder, and into the urinary bladder you have this these ducts opening. So this is the urinogenital passage in case of male. So in male, there is the passage is common for urine and sperms. But at a time, either urine or sperms can come out. So in the coming up classes, I will explain you the different regions of this urethra. So urethra is divided into 3 regions. There is this prostrate urethra near the prostate gland. Then there is membranous urethra, and then there is this lower part, penile urethra, that enters the penis or the penis. All right. So this we are going to do in the coming up classes. Now coming to the glands. What are the different glands? You have the seminal vesicle. They are in a pair. They are very important. 60 to 70% of the secretion or the semen is made by the seminal vesicle. Then you have the prostate. That is only one. That is also a very important gland. And the bulbourethral or Copper's gland. What does the seminal vesicle produce? The seminal vesicle produces the seminal fluid. Seminal fluid it makes up around 60 to 70%. Seminal vesicle secretes a viscous fluid that accounts or contributes to 60% of the total volume. 60% of the total volume of the semen. Semen is actually sperms plus the secretions of all the glands. And what does it contain? It contains 3 important substances, very important substances without which the sperm cannot be active. Fructose that provides energy to the sperms. Fructose that provides energy to the sperms. Fibrinogen that helps it to clot. And a very important substance called prostaglandins. This is very important. What are prostaglandins? They are like derivatives of arachidonic acid. And what is the role of this prostaglandins? That after ejaculation, when the sperms reach the female reproductive tract, these prostaglandins, they induce mild contractions in the uterus, so that there is this upward movement of sperms. That means sperms are forced upward towards the oviduct. You understanding? So prostaglandins are very, very important. So in the next class, we're going to continue from here, where I'm going to tell you about the accessory glands, and then we will study about penis and the different parts of the penis, and then I will explain you the process of. We will do a comparative account of the male and female secondary sexual organs and the homology between them. That is a very important question for NEET. And then we will start the process of fertilization. Okay, so stay tuned and join us same time tomorrow. Okay, and any of your doubts, you can just post. Yes, I will explain. What kind of clot you are talking about? We will discuss from here in the next class. Thank you so much for joining.